Uh, Good evening, everyone. JMitra Scientific Webinar proudly presents an enlightening session on importance of CLIA in diagnostic laboratory and the role of AMH in the domain of fertility. This webinar will highlight the advancements in chemiluminescence assay and its superior accuracy in laboratory diagnostics. Additionally, we will explore the pivotal role of the anti-Müllerian hormone, AMH in short form, in reproductive health and fertility assessment. AMH, of course, as everyone knows, is a key biomarker used to evaluate ovarian reserve, helping clinicians assess a woman's reproductive potential. It plays a crucial role in predicting ovarian responses to fertility treatments, diagnosing conditions like PCOS, premature ovarian insufficiency, and others. We're honored to have with us Dr. Debkumar Ray. He's a professor at Bardhavan Medical College as our distinguished speaker for this evening. With his extensive expertise in clinical biochemistry, we believe he will provide key insights into these cutting-edge diagnostic tools and help enhance your knowledge for use in your laboratory and your work settings. With that, I request Dr. Ray to please take the floor. Sir, it's all yours, sir. Over to you.
Thank you. So this is the first webinar from JMitra, and I am just inaugurating their webinar. And I am really having a good pleasure for this. I must be grateful to them because they have initiated such a good thing. So now let's start the topic or subject, what we are going to elaborate today. So the topic is importance of CLIA or chemiluminescence immunoassay in diagnostic laboratory and role of anti-Müllerian hormone or AMH in domain of fertility. So let's discuss what is immunoassay. Regarding immunoassay, we have to give some introduction before proceedings. Actually, whatever antigen or antibody we are going to assay or estimate, we need this kind of technique, keeping in mind the cost effectiveness. The test that uses antibodies and antigen to identify and measure the substances in a biological sample, particularly the samples like blood sample or the CSF we use most commonly. So widely applied in various fields, including our clinical chemistry, as well as pharmaceutical analysis, bioanalysis, toxicological analysis, and the environmental sciences or different uh, organic materials to be analyzed in environmental analysis. The cost effectiveness is a very important component of this assay, as well as the high sensitivity and specificity makes it a most common of choices analysis of difficult matrices. So there are wide variety of chemiluminescence. So what are the variant? EIA, that is enzyme immunoassay, radioimmunoassay, which is now not very commonly done. FIA, fluorescence immunoassay, and CLIA, that is chemiluminescence immunoassay. So what is luminescence? So I am just giving some focus on the luminescence, not in detail. So there is no scope to detail all this right now. So cold light that can be emitted at lower temperature and which kicks an electron of an atom out of its lower energy, that is the ground state level, into the higher energy or excited state. The excited state is unstable, hence the electron returns to the energy in the form of light and come back to the ground state. So this is the basic principle of luminescence. So energy in the form of electromagnetic wave, heat, friction, electric field, chemical reaction, etc., which is to be considered as the ground state, which will be transformed into the excited state. That is the luminescence. So there are different types of luminescence like chemiluminescence, photoluminescence, bioluminescence, electroluminescence, thermoluminescence, and cathode luminescence. So there are specific molecules to be used for this process of luminescence. So first one, which is not being used widely nowadays, it was AMPPD, stands for 3,4-methoxy 4-phenyl 1,2-dioxetane. So this AMPPD is a substrate for alkaline phosphatase, or in abbreviation ALP. When alkaline phosphatase catalytically hydrolyzes AMPPD, it breaks down into AMPD. And then the AMPD emits light in high intensity as a signal. Then the amount of light emitted depends on the concentration of the alkaline phosphatase. The second one after AMPPD is another luminescent, that is luminol. Luminol is a substrate of horseradish peroxidase, or HRP. Luminol reacts with the base to form the dianion. The hydrogen peroxide oxidizes the dianion. The reaction produces water, nitrogen gas, and dicarboxylate ion. And this ion emits the photon of blue-green light as it returns to its ground state. The third one is acridinium ester, which is an abbreviation AE. It's a synthetic molecule that emits light, which is most commonly being used nowadays, acridinium ester. So this acridinium ester reacts with the proteins to produce derivative that are stable and immunoreactive. And they emit light when combined with the alkaline peroxide and a cationic surfactant. So the light emission reaction is simple and doesn't require catalysis. So acridinium ester, or AE, produces much stronger chemiluminescence signal compared to luminol. That's why nowadays we are using acridinium ester instead of AMPPD and luminol, etc. Now there are different methods of luminescence. The first one is sandwich method, where the magnetic bead coated with the antibody comes in contact with the antigen or antibody as a sample. And it will again combine with the AE, or acridinium ester conjugated antibody, after a specific incubation at the specific temperature, which is 37 degrees centigrade. So magnetic separation will take place and the compound will be produced, which is called sandwich method. The second one is the indirect method, where the initial component will combine with the antibody or the sample and will produce antigen antibody complex, and which will combine again with the acridinium ester conjugated antibody will be produced after a specific incubation at 37 degrees centigrade temperature. By the magnetic separation, the compound will be produced. This is indirect method. And the third one is the competitive method. The magnetic bead coated with antibody will comes in contact with the sample which contains antigen, and it will be combined again with the acridinium ester, this luminol antigen, and after a specific incubation with the same as before, 37 degrees centigrade, the magnetic separation will produce the compound, which is the third type, which is competitive method. So the scope of the test or tests. So there are different domains we can cover. We can diagnose, we can do the follow-up for the treatment. Say, for example, thyroid. The hormonal assay we can do by using this method like CLIA or chemiluminescence method. So TSH, T3, T4, as well as free form of T3, T4, as well as anti-TPO, thyroperoxidase, and anti-TG, thyroglobulin, and thyroglobulin itself, we can assess for the diagnosis of different kinds of different variant related to the thyroid gland. The second one is the vector-borne diseases like dengue NS1, IgM, IgG for dengue, malaria antigen, leptospira IgM, chikungunya IgM, and Scrubs typus IgM variant we can assess, and we can get an idea about the vector-borne disease, and we can establish our diagnosis. And the next one is the infectious disease like HIV, antigen antibody we can diagnose, we can assay, and for the diagnosis of HIV, and surface antigen, hepatitis B surface antigen or Australia antigen, what we say, anti-HCV and anti-TP also we can assess, and which helps us for the diagnosis of infectious diseases. And apart from this, thyroid diseases, assessment of thyroid diseases or the diagnosis of thyroid diseases by assessment of different thyroid-related hormones and antigen antibodies. We can go for the fertility profile like hormones which are related to the fertility. For the treatment of infertility, we can assess such kind of hormones like FSH, follicle stimulating hormone LH, PRL, prolactin, beta HCG, testosterone, progesterone, estradiol, E2 specifically, and cortisol. So these are the hormones which is being assessed for the treatment of infertility. Now next one is the reproductive. Which one is the most common focus here for the today's discussion? That's the AMH. I'll go in detail later. And inhibin B, INH, B, PAPPD, which is pregnancy-associated plasma protein A, and ultra E3 and free beta HCG is also being assessed as a part of the reproductive system. Now the next one is the inflammatory markers. HSCRP, high sensitive C-reactive protein, procalcitonin, interleukin 6, SSA, that is serum amyloid A, and HBP is the heparin binding protein we can assess as inflammatory marker. So the most important and the most commonly done biomarkers which is associated with the cardiac diseases. These are HSCTNI, that is high sensitive cardiac troponin I, as well as cardiac troponin T. So both of these isoenzymes you can assess. Apart from this, the specific isoenzymes for CK or creatine kinase MB variant we can assay because the MB variant is present in cardiac muscles only. And myoglobulin ST2, that is suppression of tumorigenicity 2 protein, NT-proBNP or brain natriuretic peptide, LAPLA2, that is lipoprotein associated phospholipase A2, and D-dimer HFABP, that is heart type fatty acid binding protein, which is supposed to be considered as the newest cardiac marker, and ACE100 beta, that is a protein biomarker that can be measured to find out that can be measured from the CSF to find out any injury in the central nervous system or CNS. So CNS injury we can detect and we can use this kind of biomarker like ACE100 beta. Now, apart from this torch panel, we can do the toxoplasma IgG and IgM, rubella for IgM and IgG, cytomegalovirus IgM and IgG, herpes IgM type 1 and type 2, as well as herpes IgG type 1 and 2 we can assess by using this technique. Now the next one is autoimmune disease profile like anti-CCP. Anti-CCP cyclic citrullinated peptide, ACL, that is anterior cruciate ligament injury biomarker. And APL is the acute promyelocytic leukemia marker. Anti-GAD, that is glutamic acid decarboxylase having 65 kilodalton component, which we can assess by using this technique, as well as TG, TTG, and ACL70. ACL70 is a scleroderma associated autoantigen having 70 kilodalton molecular weight, and anti-nuclear antibodies also being detected. And all these are the part of autoimmune disease profile, or considered as the autoimmune disease profile or markers, biomarkers for autoimmune diseases. Now, next one is the bone markers, or which is associated with the bone metabolism or calcification of bones, like vitamin D and parathyroid hormone, as well as GH or growth hormone. Now, glycometabolism, which is related to the diabetes, insulin we can measure, as well as C-peptide we can measure by using this technique. And the last one is the tumor marker, and which is having a wide variety: alpha fetoprotein, CEA, carcinoembryonic antigen, CA125, 19.9, PSA total and free, which is in abbreviation TPSA and FPSA, and CA153. And next one is NSE, neuron-specific enolase, and PG1, PG2, beta-2 microglobulin, pro-GRP, SCCA, and CYFR21. What is the pro-GRP? That is progastrin releasing peptide, and SCCA is a squamous cell carcinoma antigen, And the last one is the cytokine 19 fragment, which is most commonly used for non-small cell carcinoma lung. So this is the biomarker for non-small cell carcinoma lung. And the last part of these components are allergy profile IgG we can assess, and anemia profile. This is not the total profile, but the biomarkers, which is most commonly being done by using chemiluminescence, that is vitamin B12, folate, and ferritin is also being assessed by using chemiluminescence technique. Now, what are the difference between what are the advantages of CLI over uh, ELISA, enzyme linked immunosorbent assay? So if you consider the accuracy, the more accurate, so I am telling about the CLIA, which is more accurate than uh, ELISA in detecting antigen and antibody. Instead of that, less sensitive. The sensitivity is less for ELISA compared to CLIA. So accuracy is more in CLIA in comparison with ELISA technique. Now, next one is the automation and time. Previously, we have an idea that if the number of samples are not inadequate, so there is no use to go for CLI. It's not like that. It is more automated than ELISA, all of you know. And ELISA is having some human error. So everything we are doing by manual method. Nowadays, I do not know whether it is widely used or not, that full automated ELISA. If it is used like that way, it must be better. So more automated than ELISA. Even in smaller sample size, it is possible nowadays. Even the sample size are very small. You can do CLI, and random use of this machine is possible. And the most important and most crucial factor here is that turnaround time is better for reporting. This is applicable for CLIA. But in ELISA, the TAT time is more because we have to do everything manually. And most commonly used semi-automated method, where we are getting a very small sample load, and the full automated system has limitation of random processing of the samples, but not like that. Right now, we can use a CLI as the random processing, and we can do even for a small volume of samples. Now, the next one is the sample volume. So in CLIA, it is very much suitable for large sample load, and even for the small sample load also. So for ELISA or enzyme-linked immunosorbent assay, large sample load is very difficult to manage or very difficult to process in a short period of time. Now the cost and shelf life. Now in CLIA, it's costlier than ELISA. There is no doubt. So chemiluminescent substrate used in CLIA often have a shorter shelf life. So that is the disadvantage of CLIA. And the ELISA, what we get advantage, it is cheaper than CLIA. So colorimetric substrates have been have a better shelf life. So shelf life, if you consider, the shelf life is better in ELISA, but it is not acceptable for CLIA. But right now we can use CLI must use to get more accuracy, more precision, and better result, and we can minimize the TAT time. Now the second part of our topic is the importance of AMH or anti-Mullerian hormone. So basically, we should explain few things which are very important here. These are the genetic aspect. The gene for AMH or anti-Mullerian hormone encodes a 560 amino acid protein and whose carboxyl terminal domain shows marked homology with a transforming growth factor, which is TGF beta variant, and the beta chain of inhibin and activin. The gene has been localized to the short arm of chromosome 19. So AMH is secreted mainly from human fetal and postnatal Sertoli cells, and until 8 to 10 years of age, and can be used as a marker for the presence of these cells. So there are some specific components or gene, that is SF1, WT1, SOX9, and GATA4 has been shown to regulate AMH gene expression. So the human AMH receptor gene has been cloned and mapped to the Q13 band of chromosome 12. The receptor has been identified as being similar to other type 2 receptor of transforming growth factor beta family. The next one, the AMH, which stands for anti-Mullerian hormone. So it is having the 140 kilodalton molecular weight, homodimeric disulfide linkage, glycoprotein in nature. So it is a glycoprotein hormone, and the TGF transforming growth factor beta superfamily, what I have mentioned in my previous slide. So also known as Mullerian inhibiting substance or MIS. So both of these are synonymous and plays predominant role in male and female sex differentiation. In males, it is expressed by substanticular, which is Sertoli cells. Similar, substanticular cells or Sertoli cell in the testes and promotes the regression of Mullerian duct and help in development of male reproductive system. So this is very important for sex determination. In female, it is expressed by granulosa cells of developing pre-antral and antral follicles. Now, the differentiation of Wolffian duct is mediated by testosterone secretion from the testes. All of you know. The SF on the specific gene regulates steroidogenesis, that is the synthesis of steroid hormones by Leydig cells in the testes by binding to the promoter of genes encoding P450SCC and P450C17. The presence of an ovary or in the absence of the functional fetal cells, fetal testis, Mullerian duct differentiation occurs and the Wolffian duct involute. So these are the genetic aspect of and control or regulation of AMH. Now this AMH or anti-Mullerian hormone. So this is the graphical presentation. From the fetal stage to the early adolescence, the AMH levels are relatively high and then slowly decreases from the age 25 or 25 years. And then longitudinal decline of is seen and becomes undetectable at about 5 years prior to menopause. So in the lower part, what I'm just a minute. So these are the anyway. This AMH secretion for female, it is going to start at the level of 36 gestational weeks, going up, and it will be getting its peak at the puberty. And instead of that, for male, it will be maximum in 8 gestational weeks, and it will going to decline. And in adulthood, it will be very less, and it will be continued in a sustainable level up to death. And but in case of female, it will decline and combines the baseline or comes into the baseline. And the next one is, what about the physiology of anti-Mullerian hormone? So in association with the testosterone, mesonephric or Wolffian duct develop into epididymis, vas deferens, seminal vesicles, part of male reproductive system, while paramesonephric or Mullerian duct regresses. So these are the component of AMH, but the AMH is increases. Now AMH is going down in absence of testosterone, and AMH paramesonephric ducts develop into the fallopian tubes, uterus, and proximal vagina, parts of female reproductive system, and mesonephric duct regenerate. So these are the way by which the sex determination is being done by the direct effect of AMH. Now, AMH dynamics or anti-Mullerian hormone dynamics in females. What is happening? Both AMH and primordial follicles decreases age after sexual maturation, and which indicates that AMH is a marker in ovarian age detection. So we can use AMH assessment or assay of AMH by using CLIA or ELISA, preferably CLIA, to detect the ovarian age. So in x-axis, what we are showing here, number of primordial follicles is going to be increased, and as age advances, the AMH level will go down. And these are the years we have given in the y-axis. Sorry, in x-axis we have given the years. After the 48 years, nearly the menopausal age, it is going down, and it coincides with the baseline And instead of that, this part, the AMH is uh, going up in its normal consequences, which is not at all related to the number of primordial follicles, which is being uh, associated with the AMH variations. The next one is AMH anti-mullerian hormone, the clinical implications. The assisted reproductive technology always uses AMH assessment or assay of AMH, and infertility preservation also needs this one. And ovarian neoplasm is also be detected by the assessment of AMH. And polycystic ovarian disease like PCOS, previously it was PCOD. Polycystic ovarian disease is also associated with the assessment of AMH. Pediatric reproductive endocrinology needed the assessment of AMH, and prediction of early menopause is also being done as because it makes the uh, ovarian age. It can assess the ovarian age. That's why early menopausal can predict. And the next one is the AMH. Uh, Levels do not vary throughout the ovarian cycle of women. And estradiol, inhibin A and B of two types, luteinizing hormones or LH, follicle stimulating hormone FSH levels, all of the hormones, what we are showing here, starting from the top, that is estradiol, what we express in terms of picomole per liter, and inhibin A and B, luteinizing hormone and FSH, there are the variations. But what we want to focus here, this is the line which is for AMH, which is expressed in terms of very minute quantities as picomole per liter, which is remain constant. So that's why we have written here all hormonal level will vary, changing throughout the different phases of the ovulation or ovarian cycle, like early follicular phase, late follicular phase, periovulatory phase, mid luteal phase, and end luteal phase. But this is constant. What I'm saying, AMH level remain constant, this blue line. The next one, this is the last slide probably. So this is the pie chart, where we are showing the path uh, to childhood. This one is the puberty, this one is the adulthood, and what are the role of AMH in birth and childhood? The differential diagnosis is being met, and disorders of sex development are also being assessed by AMH. The second one is the pre-puberty. So differential diagnosis of constitutive uh, pubertal delay and congenital hypergonadotropic hypergonadism like CHH we can detect. And AMH is also important in adulthood, where you can assess the ovarian reserve assessment, prediction of menopause or ovarian age. We can detect by using this assessment of this hormone like AMH, and other fertility related diseases. And for the treatment of infertility, we can assess this one. And application in assisted reproductive technology, there are widely it is being used widely. Now, potential diagnostic marker for polycystic ovarian disease or PCOS, and fertility preservation programs in oncofertility is being done by this assessment of such a hormone like AMH. And lastly, screening of ovarian granulosa cell tumor is also being established by assessment of AMH. So these are the entire part of the uh, uh, topic. And thank you all for your presence. And
Thank you very much,
thank
doctor.
you to JMitra for their initiative, for such kind of good initiative. I would like to expect such kind of initiative in future, in near future,
Absolutely.
repeatedly. Thank you.
So there was one question, if we may trouble you with that question, uh, from one of our attendees who asked, which detection method is superior in CLIA? Is it flash CLIA or glow CLIA?
See, we are so CLIA we have mentioned here, but there is limited scope to say everything about the different variants of CLIA. Grossly, we can say the flash CLIA is the more effective method if you focus on the particular subvariant of CLIA, there are different type of CLIA. We are not focusing. We have a limited scope to discuss all this in this discussion. So that's why I would like to say you must prefer the first one.
Right, sir. And well, thank you very much for your time, sir. We have a small certificate of appreciation from our end, Dr. Debkumar Ray, for presenting this wonderful scientific lecture on importance of CLIA in diagnostic laboratories and the role of AMH in the domain of fertility. Of course, the hard copy of the certificate will be delivered by one of our field salespersons to you directly, the manager most probably, and we'll have that sent across to you after getting it framed, sir. But please accept this digital offering for now. We'll have the hard copy sent to you soon. Thank you very much, Dr. Ray, and we look forward to collaborating with you in the future on further academic sessions, sir. Thank
I
you.
am very much glad to receive your compliment in the form of digital format, as well as
Thank you,
what
sir.
you are saying later I'll receive. Thank you. Thank you so much.
Thank
Thank
you
you.
very much, Doctor. And thank you all, ladies and gentlemen, for joining us today. We'll be back with another webinar very shortly over the next 2 weeks. Thank you and have a great day. Jai Hind.